Welcome to G Fun Facts Online. Imagine holding a fossil, a remnant of a creature that lived millions of years ago. For centuries, we've studied their shape and size to guess at their story. But what if we could make that fossil talk? What if it could tell us its genetic secrets, its sex, its exact place in the family tree? That science fiction scenario is now a reality. In paleontology, a revolution is underway, and it's all thanks to a field called paleoproteomics. Scientists are now pulling ancient proteins from the one part of the body that can survive millions of years, tooth enamel. This isn't just a small step forward. It's like we found a biological Rosetta Stone, allowing us to read the lost language of deep time. So how are the hardest tissues in our bodies revealing the softest, most detailed secrets of our ancient past? Let's find out. The remarkable ability of tooth enamel to preserve these biological clues for millions of years lies in its unique structure. Enamel is the hardest substance any vertebrate makes, composed of about 96% inorganic minerals, mostly hydroxyapatite crystals. Think of it as a biological ceramic. During its formation, a set of specialized proteins act like microscopic construction workers, organizing these crystals into their incredibly dense and durable structure. As the enamel matures, most of these proteins are cleared out, but tiny fragments get trapped inside the crystalline matrix. This mineral fortress acts as an indestructible time capsule, shielding these proteins from water, microbes, and temperature changes that would destroy other organic matter. As Dr. Daniel Green, a researcher at Harvard and Columbia, puts it, "Teeth are rocks in our mouths." They're so tough that a tooth that is 100 million years old can still contain a geochemical record of that animal's life. While bones also contain proteins like collagen, enamel is special. The set of proteins in enamel is relatively small and unique to that tissue, which makes analysis much cleaner and reduces the risk of contamination from the soil the fossil was buried in. For decades, the gold standard for peeking into the past was ancient DNA, or aDNA. It completely redrew the human family tree, revealing lost relatives like the Denisovans, and showing how our ancestors interbred with Neanderthals. But DNA is fragile. It breaks down quickly, especially in the heat. Outside of the frozen permafrost, it's almost impossible to recover aDNA from fossils older than about 500,000 years. This leaves a huge gap in our knowledge, because so much of human evolution happened in the warmer climates of Africa and Asia. This is where paleoproteomics becomes a superhero. Proteins are simply tougher than DNA. The chemical bonds holding them together are more robust. And when they're locked inside the mineral vault of tooth enamel, they can survive for an astonishingly long time, far beyond the limits of aDNA. In 2019, researchers proved this by sequencing proteins from a 1.9 million-year-old molar of Gigantopithecus blacki, a giant ape from a subtropical region of China, an environment where aDNA wouldn't stand a chance. Shortly after, they analyzed a 1.77 million-year-old rhino tooth from Dmanisi, Georgia. These studies smashed through the aDNA time barrier, but the story gets even crazier. In 2025, two studies blew all previous records away. One team recovered protein fragments from rhino and elephant teeth in Kenya's Turkana Basin that were between 1.5 and 18 million years old. Another study found proteins in a rhino relative's tooth from the Canadian high Arctic, dating back more than 20 million years. This firmly established proteins as the most durable source of ancient genetic information we have. The secret to this power lies in a small but highly informative group of proteins. The most important of these is called amelogenin. Amelogenin is the main protein used to build enamel, but its real value for paleontologists comes from a genetic quirk. The gene for amelogenin is located on both the X and Y sex chromosomes. This means there are two slightly different versions of the protein, one for the X chromosome and one for the Y. By using their high-tech equipment to find the Y chromosome-specific version of the protein, scientists can definitively say a fossil belonged to a male. If they only find the X chromosome version, it's a female. This is a massive breakthrough. Determining the sex of a fossil from bones alone is often impossible, especially if you only have a few fragments or the remains of a juvenile. This one protein gives us a direct, unambiguous answer. Beyond amelogenin, other proteins like ameloblastin and enamelin also get trapped. Since the blueprints for these proteins are written in an organism's DNA, their amino acid sequences are unique to different species and evolve over time. By comparing the ancient sequences to those of living animals, we can reconstruct evolutionary family trees with incredible accuracy. So how exactly do scientists perform this molecular magic? The process is delicate and requires ultra-clean labs to avoid contamination from modern proteins, like a flake of a researcher's own skin. It starts with carefully drilling a tiny amount of powder from the fossil tooth's interior. This powder is then treated with a weak acid, which dissolves the mineral crystals and releases the trapped protein fragments. The mixture of ancient protein fragments, called peptides, is then injected into the heart of the laboratory, a machine called a liquid chromatograph tandem mass spectrometer, or LC megasiemens per megasiemens. First, the liquid chromatograph separates the complex mixture of peptides based on their chemical properties. Then, the mass spectrometer does two things. It weighs the intact peptides, and then it selects them one by one, smashes them into smaller pieces, and weighs those fragments. The result is a massive data set of fragmentation patterns. Bioinformatics software then acts like a detective, comparing these patterns to a database of known protein sequences from living species. By matching the fragments, the software can identify the original peptide and piece it together with others to reconstruct a large part of the ancient protein sequence. A final crucial step is authentication. How do they know the proteins are truly ancient? Over millions of years, certain amino acids degrade in predictable ways. Finding these specific patterns of chemical damage is like finding a made in the Miocene stamp, confirming the protein's antiquity. This incredible technique is already delivering stunning results that are rewriting textbooks. For years, the giant ape Gigantopithecus blacki was a mystery. We only had its teeth and a few jawbones, and no DNA could be recovered from its warm, subtropical home. In 2019, the analysis of proteins from a 1.9 million-year-old molar solved the puzzle. The sequences showed that Gigantopithecus was a close relative of the modern orangutan, with their family lines splitting about 10 to 12 million years ago. The technique has also given us a closer look at our own family. In 2020, protein analysis of an 800,000-year-old Homo antecessor fossil from Spain placed it as a close sister lineage to the common ancestor of modern humans, Neanderthals and Denisovans, clarifying its spot in our family tree. Even more amazing was a 2025 study on 2-million-year-old fossils of Paranthropus robustus from a cave in South Africa. This was a distant, robust-jawed cousin of early humans. By analyzing the amelogenin protein, the team determined the sex of 4 different individuals, 2 males and 2 females. This was the first time direct molecular evidence had ever been used to sex such ancient hominins. But it doesn't stop with primates. A 1.77 million-year-old rhino tooth from Dmanisi, Georgia, helped solve a long-standing debate. The protein evidence showed that the famous woolly rhino evolved from this earlier Stephanorhinus lineage. And remember those record-breaking fossils from Kenya's Turkana Basin? Protein surviving for 18 million years in one of the hottest places on Earth proved that this technique has an incredible future. The fragmented sequences were still clear enough to link a 16-million-year-old elephant relative to modern elephants, and an ancient hippo to today's hippos and their closest relatives, whales. Now, despite its revolutionary power, paleoproteomics has its limits. The biggest one is the amount of information you can get. The enamel proteome is based on just a handful of genes, compared to the tens of thousands in a full genome. So we can't do things like calculate the exact percentage of Neanderthal DNA in a person, something aDNA is great for. Also, while proteins are tough, they do still break down over millions of years, meaning we only ever get fragments of the original sequence. But the future is incredibly bright. Mass spectrometers are becoming more sensitive, able to detect even smaller amounts of protein. Scientists are pushing the time limits further and further. The discovery of 20-million-year-old proteins has moved the idea of recovering proteins from dinosaurs out of pure science fiction and into the realm of plausible scientific inquiry. Researchers are also exploring what else these proteins can tell us, like revealing clues about the health and physiology of an ancient animal. In conclusion, tooth enamel proteomics is a monumental leap forward. It has broken through the time barrier that held ancient DNA research back, opening up millions of years of evolutionary history for direct genetic study. It's allowing us to determine the sex of our 2-million-year-old relatives, redraw the family trees of extinct giants, and prove evolutionary links that were once just educated guesses. The silent stones and fossilized teeth of our planet's deep past are finally beginning to speak, and they are telling us the most profound stories of all, the story of life, and our own origins within it.